Hi, everybody. Thanks, Idan, and thank you, everybody, for this great meeting. I'm going to discuss today our findings related to the SHANK3. The presentation is focused on the interplay between neurons and glia, which is something that in autism field we less study. So our lab is focused on the interplay between neurons and those who cover neurons with myelin. So these are the oligodendrocytes, which in autism less people are interested and focused on. And this interplay between neurons and glia affect the axonal conductivity that might affect neural circuits synchronization, which is critical for behavior outcomes. And in autism specifically, we study these interplays. And unfortunately, white matter is much less studied in aspects related to social neuroscience, and I want to emphasize that this is important and convince you of this relevant aspect in autism. So here we can see many brain regions which are highly active during social behavior in humans. And if I look at those brain regions, they are interconnected with white matter, meaning axons covered with myelin that is critical for the synchronization of this social brain and other brain regions. And we are interested in myelin. And actually, if you look at this Nature Genetics paper, you see here many genes related to autism, heavily involved in autism. And actually, if you look at them deeply, 33% of those genes are actually related to the regulation of oligodendrocytes, those cells that make myelin, their development and function. And one of those genes is SHANK3, which is going to be the focus of this talk. Recent studies show that SHANK3 mutation or deletion leads to white matter abnormalities, but the information was very limited, suggesting thinner corpus callosum, which is heavily myelinated, and only demonstrating that SHANK3 is expressed in those cells that make myelin, these myelinating oligodendrocytes. So we wanted to further study SHANK3, which, as you can see, is highly involved in autism, but was mainly studied in neurons. So in this synaptic aspect, synaptic compartment here at the postsynaptic site between neurons, SHANK3 was heavily studied for its function in holding those NMDA receptors and others. But not only in neurons, SHANK3 has roles, as you will soon see. So our study is based on a mouse model that was developed at MIT, in which there is a specific insertion of guanine in locus 3680 in SHANK gene, leading to an early stop codon that leads to the reduced expression of SHANK3 in Phelan-McDermid syndrome and autistic features involved. And this mouse was heavily studied and characterized for repetitive behavior, abnormal social behavior, abnormal physiological aspects related to neurons, which again was the main topic related to SHANK3. And this mutation affected not only the physiological function, but also reduced the expression of other postsynaptic density proteins and those receptors. So while this was heavily studied and published already, in Bar Fisher from my lab, which is here actually, wanted to study whether SHANK3 has a similar role, but not in the communication between neuron and neuron, but between a neuron and an oligodendrocyte. So whether there is a SHANK3 role for a postsynaptic site in oligodendrocyte precursor cells. So we all know about neuron-neuron interaction, but as you can see here, there is also a synapse between a neuron axon and oligodendrocyte precursor cells, OPCs from now. And this communication is critical for the development of those cells into mature myelinating oligodendrocyte. And if we zoom in into this synapse, you see the axon here, and you see the OPC, which holds receptors in its membrane critical for the physiology of OPC and the influx of calcium, which is critical for the formation of myelinating oligodendrocytes. And previous studies showed that OPCs have synapses and that those synapses have postsynaptic sites, including Jefferin, which is a postsynaptic protein. So this was already showed, but not necessarily in the frame of SHANK3. So in my lab, we established a primary culture in which you have highly enriched OPCs, oligodendrocyte precursor cells, labeled by PDGFR alpha. And if we compare between the control and the mutant cells, you see that SHANK3 is indeed expressed in OPCs and is reduced in those OPCs when you have the mutation. This leads to reduced expression of the protein SHANK3 in those OPCs, leading to the reduction in other genes related to proteins encoding proteins that are in the postsynaptic site and NMDA receptors and others. Not only on the molecular aspect, also on the cellular aspect, you see that the differentiation of these precursor cells into myelinating oligodendrocyte is actually impaired, and the expression of the myelin basic protein in those cells is affected, leading to abnormal development of these myelinating cells due to histone modifications that affect their differentiation. But the question is whether this affects something physiologically. So we induced electrophysiological signals by applying glutamate on this primary culture and quantified using fluo4 the calcium activity. And what you can see is that when you have mutated SHANK3 in OPCs, you have abnormal calcium influx, leading to lower activity rate in those OPCs with the mutation, affecting their amplitude and the inter-event intervals and other physiological aspects. Not only the calcium influx was affected, but also electrophysiology. When we patch clamped those cells, we saw abnormal sodium and potassium currents. So moving from the in vitro level to the in vivo level, we looked at cellular properties in the mouse brain, and we saw some alterations, not very dramatic, but we wanted to ask whether the myelin fraction itself from the brain of the mouse is affected. So we biochemically enriched myelin. Instead of having whole homogenate, we were able to increase the expression of MBP by purifying myelin biochemically. And looking at the myelin fraction, we saw reduced expression of the protein level of SHANK3 and other postsynaptic proteins which are expressed in myelin and are abnormally expressed due to the mutation. Not only those proteins were affected, but also the main proteins that make myelin are actually in lower expression due to the mutation. So if we have lower levels of myelin, it might affect the white matter, as we can image by running MRI diffusion tensor imaging. And indeed, when we did this, we saw whole brain connectogram, which you see here, affected, suggesting white matter tracts are abnormally active in those brains, so that when you look specifically at the corpus callosum, which is heavily myelinated, you see that the number of fibers in the mutant mouse is actually affected, and the white matter properties are abnormal. Not only the white matter level is abnormal, we also looked at the ultrastructure level of the myelin by using EM, electron microscopy, and we saw that on the ultrastructural level, the axon diameter of the mutant mice are lower, and there is increased number of myelin deformation, meaning that the compaction of myelin is impaired when you have a mutation in SHANK3. So if the structure of myelin is abnormally expressed, it will lead potentially to physiological aspects related to axonal conductivity. And we measured that in the corpus callosum of mice in an ex vivo formation. And what we found is that the N2 signal was higher, suggesting that there is more conduction on unmyelinated exons, indicating physiological deficits in white matter. So all of this is coming from a mouse model with a human mutation, just like in humans, but we induced it in mice. But to validate that these are indeed the phenotypes, also in more clinically relevant samples, we took fibroblasts from a girl from Israel who has the same mutation by chance. And when we did this, we reprogrammed those cells into stem cells and then differentiated them into oligodendrocyte precursor cells. And when we measure the physiological aspects of calcium influx in those cells, in those OPCs coming from the patient and her mother, we were able to see the same phenotype as we characterized in our mouse primary culture, but now in human samples, validating that calcium influx in OPCs with the mutation from a patient is indeed impaired. So after we validated our findings in mice and humans, our last approach was to try and improve physiological properties of these OPCs. So what Inbar did is she tried to rescue, by a genetic treatment, rescue the expression of SHANK3 into those OPCs, coming back again to mouse primary culture. You see here cells in vitro, which are OPCs, and they don't express SHANK3 because they come from the SHANK3 knockout mouse. But with a genetic restoration in which Inbar encoded the SHANK3 human gene, full-length gene, she reintroduced SHANK into those SHANK3 knockout OPCs, which now you see that SHANK3 is expressed in those cells and in the right location, forming those synaptic butons that you see here all over the cells. Now the question is, if the restoration of SHANK3 in vitro, which now we see normal level of SHANK3, will enable restoration of other postsynaptic proteins. So Inbar measured whether PSD95, which is critical for the formation of this postsynaptic density site, also in OPCs, whether the restoration of SHANK3 will restore PSD95. And indeed, you can see that SHANK3 is expressed in those SHANK3 knockout OPCs in yellow, which enabled PSD95 now to come back and have higher levels significantly compared to non-transfected knockout cells, meaning that the formation of SHANK3 enabled proper restoration of PSD95, which is great. And now the question, if this improved postsynaptic site properties will result with improved receptor compartment properties. So Inbar characterized the NR1 subunit of the NMDA receptor, and she saw that when you restore SHANK3 in those knockout cells, the expression level of NMDA receptor is now higher compared to untreated knockout cells, meaning that this genetic restoration postnatally is effective in those properties of the synapse. So to summarize, what we show is that SHANK3 is critical in OPCs, a less studied aspect in autism, and is critical for the holding of these receptors, critical for the entry of ions, critical for the transcription of mRNA and proteins translation, affecting cellular properties, ultrastructure of myelin, and white matter properties. And when you have the mutation, as in Phelan-McDermid syndrome, you will have reduced amount of receptors, reduced ion influx, affecting the translation and transcription of genes and proteins, which will affect the properties of myelin and white matter. So with that, I would like to conclude and thank the lab members, and especially Inbal and Sophie, which are here, and Inbar is also sitting here, and collaborators. This was done with a great collaboration with Shani Stern from Haifa University and Ben Maoz from Tel Aviv University, our funding, and you guys. Thank you.